Hello, and welcome to today's webinar, Engineered Particle Platform for Rapid Advancement of Inhaled Formulations, presented by Inhalation Magazine and sponsored by Capsugel. My name is Elisa, and I will be the moderator for this webinar. Today, our presenters will be Devin DeBose, who is the head of Inhalation Product Development at Capsugel Bend, and Philip Kuehl, scientist and director of Scientific Core Laboratories at Lovelace Biomedical. Before we begin, I'd like to remind our viewers that we will hold a Q&A session at the end of the presentation. You could submit a question at any time using the Q&A box on your screen. Now, Devin, I will pass it off to you to begin.
Thank you, Elisa. So we have both Phil and I from two different organizations today, which I think provides a unique opportunity for us. This is kind of a culmination of about a 15-year collaboration. And I think provides a unique opportunity with some of the in vivo data that we can actually share publicly that Phil will go into. So we have a lot to go through today, so I'll jump right into it. So what we'll show is a spray drying and an encapsulation platform interspersed with some of the in vivo data and in vivo approaches that Phil will go through. And I'll pass it off to Phil for this section.
All right, so to begin with, what Devin said was I think probably really important. What we're presenting today are some case studies with some real data on some problems that the teams at Lovelace and Capsugel have worked together to address and solve. By no means is this meant to be an all-inclusive, one-size-fits-all approach to anything. As any of us know, there are multiple different delivery routes for inhalation. And within those delivery routes, within nebulizers, DPIs, MDIs, there's different formulation techniques for each of those that can help to be used to address problem statements or different situations within a program. What we're hoping to do here is to show you some examples, some strategies that have been used, give you an opportunity to ask questions on those, and really be able to show the focus kind of moving down in through the right-hand side of your slides, talking through the ability to look at lung disease or clinical indications and how to translate that through a formulation into an animal model to test efficacy, ultimately doing what we're all hoping we're able to do, which is address a clinical need or a therapeutic indication that has unmet needs or opportunities for the health within the populations.
Thanks, Phil. So before we start, if we wanted to cover kind of what are some trends that we're seeing, what kind of problem statements are our clients bringing to us? So a lot of these things we're seeing new compounds, NCEs that aren't always compatible with traditional delivery techniques, things that don't mill well, maybe things that are amorphous, stability or solubility in water or HFA-based carriers, and certainly with the NCEs, the need to have drug-sparing capabilities for feasibility and scale-up. So, working with milligram-type quantities early on, coupled with low delivery efficiency, especially in toxicology studies, which Phil will go into a little bit later. And also maybe more uniquely is tunability of the PK and or stability needs based on amorphous or a crystalline format. A lot of people think of spray drying as an amorphous format, which we'll talk through some ways of where maybe we can change that. Some additional market research here that we'll just go through quickly that I think is worth noting. So a majority of new development programs are, in fact, dry powder inhalers, shown on this slide, and also, shown that the majority of them are small molecule. But I think equally unique and in line with what Phil covered is a lot of these different individual molecules may have, 2 or 3 different delivery techniques throughout their lifetime. So, shown here is some have a nebulizer, MDI, or DPI approach at some point in their lifetime. So how have some of these issues been addressed for some of these complex molecules? Well, there's certainly lots of different technologies out there. Some are spray drying, some are not. Some of these are shown on the left-hand side. You can see some of the unique different particle morphologies. And we'll kind of show some similar approaches, and in some cases, some unique approaches that we've developed to help deal with some of the more complex molecules or approaches that are coming through different drug delivery pipelines. Slide 8 just provides a kind of an overview or a map of what we'll use throughout this presentation and revisit it at key points. But starting off with kind of more just the problem statement definition. So a lot of different things need to be considered early on to kind of ensure a streamlined drug development process. What's shown here is just different considerations for, what's the product profile. A lot of people are starting to look at the oral BCS and trying to define how to use that for inhalation drug delivery. So, if I'm crystalline or amorphous, will that impact PK? Where is what's the mechanism of my drug and ultimately the site of action? Phil will talk about these two particular things in the upper right-hand corner in much more detail in some of his interesting in vivo studies that we've collaborated on. Similarly, with different drug properties, how can we use those to our advantage? Is an amorphous approach acceptable from a physical stability or from a PK perspective? Taking things like the glass transition temperature and melting temperature, desired dose. What does that mean from an active loading? What does that mean from the actual solubility in the lung fluid? And can we impact that through either formulation or the spray drying process to our advantage? And we'll talk through some approaches there. So using the same map and just jumping into kind of the formulation and process side of spray drying. So what's shown here is just a very simple spray drying overview. Most of you are probably familiar with it, at least to this detail. We're taking, drug, excipient, dissolving them in a spray solution, feeding them to an atomizer, which is a very critical part of the process, as this ultimately defines the particle size, drying those droplets down to a particle, and then the collection, usually via a cyclone separator, another key part of the process that sometimes needs some additional engineering to optimize to collect these very fine particles. But kind of the API properties, excipient properties, and process can be used to manipulate the final product properties, to our advantage. Sometimes an amorphous formulation works great, sometimes it may not. Pulmonary excipient selection. Ideally we don't want to add anything. But in certain cases, maybe due to dose dilution or just for aerosolization of what I would say are generally very poor flowing, poor handling powders, we might need to have some sort of surface modification to help improve dispersibility. So there's certainly options out there. The following slide shows a little bit of how limited we are. So these are different excipient approaches out there. It shows a variety of different purposes, but interestingly, you see the things in bold, which may be difficult to see, is the list of actually approved excipients and dry powder inhalers. So probably of all the drug delivery routes, this one actually has the most limitations from an IAG standpoint. So just a simple approach. Leucine has been around in the literature for quite a while, and I feel like you can't really see a new paper on DPI work with spray drying without leucine being involved. So just kind of showing here, how it's used. On the left, it's just a higher molecular weight PDP, kind of mimicking a protein, and on the right, we just have a trehalose leucine formulation. You can see the increased rugosity and wrinkled morphology, which minimizes surface interactions and generally leads to improved dispersibility of the powder. So a pretty common technique. And been around for quite a while now, and we'll show some examples of using this in a simple format. Water uptake. This is something we'll revisit throughout the presentation as well. Generally, with spray drying, and especially with some of these sugars that might be used in the formulation, they're rather hygroscopic. So water content can be very important to consider throughout the process, both in formulation selection, process, handling, storage, etc. So what's shown here, and this is what we'll commonly look at for different formulations, is the glass transition temperature versus relative humidity, as well as water uptake measured via DBS. So you can see for some of these simple sugars like trehalose and lactose, where you have a high initial glass transition temperature dry, but the the glass transition temperature would depress significantly with different amounts of water, and at some point ultimately crystallize, which usually will lead to massive fusion events and no longer have a powder suitable for inhalation. So a little bit more on just the spray drying process background. We have basically these droplets being introduced into the spray dry chamber, the heart of the system, where we're basically contacting these particles with hot drying gas almost immediately, forcing very rapid solidification and evaporation to the dry state. This can be very important for things like biologics, where people might look at the high temperatures and think they can never survive that. The nice thing, though, is we have evaporative cooling at the surface, which actually takes and shields the particles from the high temperatures of the process and the rapid solidification, hopefully in a high glass transition temperature matrix, to limit mobility of these particles as they're swirling throughout the chamber. What's shown here is just some simple offline tools that we've developed, especially when we're working on an NCE where we're bulk limited. We can kind of use offline tools like a phased Doppler particle analyzer to understand the atomization. Tools like a hot bench or hot plate experiment to understand what kind of thermal constraints are in the system. Where should we limit our inlet and outlet temperatures? Things like dynamic vapor absorption I showed earlier to find what kind of outlet relative humidity can I run at and what should be my target water content, and kind of can use these to develop these thermodynamic maps to utilize these offline results to define how we do the spray drying process. So I just want to show kind of, when using these tools, how we can have a bulk sparing approach. So what's shown here is a simple albuterol sulfate water-soluble formulation with leucine. And what I'm showing here is a couple different scale spray dryers we have. So on the left is just our BLD35 lab scale with a batch size of about 300 milligrams. Fairly high yields for a batch size of 300 milligrams. And it's taking that into the same scale spray dryer with the larger batch size as well to a larger PSD1 scale spray dryer with a much larger batch size. It's showing that we can maintain the same physical properties across these scales from 300 milligrams to hundreds of grams or even kilo quantities of spray drying is a continuous process. And in many cases, a PSD1 might be a commercial volume spray dryer for smaller products. Similarly, taking advantage of a formulation like Leucine, which modifies or kind of controls the surface morphology, we can very active loadings and kind of have almost an active independent platform in certain cases, and certainly with water-soluble compounds where across active loadings and again across batch sizes at scales, we get very similar yields, water content, and aerosol properties. Similarly, the morphology shown there of the different loadings and different scales, as well as the geometric particle size. So pretty simple case study just to kind of highlight that the spray drying process can be well defined and easily scalable from both small batch size and to larger quantities. Again, revisiting water content. I talked about the formulation and excipients. If we go through the process of picking our excipients and understand the water content after spray drying, we need to pick the relative humidity that hits that desired water content from a glass transition temperature that gives us a physically stable product. And so a lot of cases this is done with either local control shown in the picture here or with sweep control. Typically, relative humidities of 30% or lower is what might be required in the spray drying process and handling using hydroscopic amorphous materials. So what if the compound isn't a simple water-soluble molecule? Certainly, it's not always as easy as what was just shown above. What's kind of shown here is a formulation map of different molecules out there with water solubility on the x-axis and the melting temp to glass transition temperature ratio, which is kind of a measurement of propensity to crystallize. And in some of these cases, when we're not in this highly water-soluble, low propensity to crystallize regime, we might have to use different approaches to help process the materials and B, drive the final product to a desired state, whether for PK or for physical stability purposes, which we'll show a couple examples. So one of those is just a simple inline mixing approach. So, we've used this to, either deal with compounds where we want to take advantage of a nice surface modifying excipient like leucine, or, with a compound, or coupled with a compound that might have water instability issues. So it's just simply taking the API in an organic solvent, something like leucine and water, feeding these to the spray dried chamber, and you'll see the note distance controlled there. In certain cases, we can actually use this to drive crystallization with the solvent and anti-solvent blends, which is what's shown in the pictures on the right, of we actually ending up with these crystalline materials out of the spray drying process, sub 5 microns. And in this case, we actually utilize leucine in a slightly different way to actually coat these spray dried crystals, which is what's shown down below to give basically a much better physical stability profile. So without leucine, we see the growth of the crystals with time, and with leucine, it's a very stable particle size. So kind of using leucine in a slightly different way in this instance. One other approach, and certainly we use this for a lot of the brick dust compounds, the compounds that are basically insoluble in just about everything, where we'll take a slurry of active and or excipient, and under high pressure feed them to a heat exchanger just before the spray drying chamber, where it will heat up that solvent to superheated temperatures under pressure to prevent boiling, where we can flash the material in and actually use now take advantage of the system seeing ambient pressure in the spray drying chamber to actually drive atomization to get a much smaller particle size. This can be used for throughput enhancement, but also, again, playing advantage of concentration over solubility ratios to drive crystallization in the droplet dry into a particle, which is shown with some of those particles on the right, where you see the more crystalline morphology with these particles embedded in some sort of matrix like trehalose or lactose, and with an overall particle size of less than 5 microns. Some other things that we're trying to look at is there's lots of different spray drying chambers out there in sizes, and a lot of them are kind of meant for multi-use. What I mean is that can serve both inhalation or oral purposes with the correct atomizers or cyclones. But for inhalation purposes, we really don't need these large spray dry chambers. These things, these very small droplets we have to produce to make these very small particles, really don't need a massive 2 or 3 story spray dryer at the end of the day, like you might need for oral formulations or for the food industry. So rather than build a $35 million commercial facility that takes 2 or 3 years to build, try and allow for more modular design both in the facility and the spray dryer. Allow for things like supply chain increases or reductions, which are both possible as prediction change. Minimize the scale of changes. Go from your 200, 300 milligram batch to actually your tens of kilos, hundreds of kilos batch on the same spray dryer. Just a couple of things that we're trying to work on at the bin site. What about for intranasal delivery? Can we also use spray drying for these purposes? Certainly, although in these cases we're generally targeting a much larger distribution where we're trying to minimize the amount of particles that make it into the lung, usually for perhaps toxicology purposes with excipients that are approved for intranasal but not for inhalation. So here's where we actually do need the larger chambers. If we need to now dry particles in the 20, 30, 40 micron range and purposely minimize fines, we have to make much larger droplets which require a much longer residence time to dry. And so here's just some of our chambers modified, to allow for some of these types of formulation. I believe I'll pass it over to Phil now to talk about how we've utilized some of these approaches to make supplies that Phil's been able to do some unique things in vivo.
So yeah, Devin, thank you for that. And what I'd like to go through now are just some of the thought process that you go through in a selection of an in vivo model, and how you can work to merge those things together with your formulation, and how you can adapt in vivo systems to make nearly any type of inhalation product work in one of those settings. So, as Devin said when he highlighted this slide, I'd like to come back to this, is what I'd like to focus on is really that top right corner of this slide, looking at what happens to a material when it deposits, and really making sure that in designing a study, you're setting yourself up to answer the right question with the right caveats to everything that's going on. And that transitions exactly into this slide, which is one of my favorite slides. I think this is something that we often forget when we're doing biomedical research, regardless of where we're delivering the material, whether it's an inhalation study or an IV study, in that all of our models are exactly that. They're models. In anything we do, we have to make sure that we're setting things up to answer specific questions so that the data and the model are useful. But in looking at this from an inhalation perspective, appreciating these differences in anatomy between the different species that we often use, and the case studies that I'm going to show are going to be rodent and dog studies today, there's a marked difference in anatomy and in all sorts of features of the delivery between humans and these models. And you have to always keep them in mind when we're working through any of these studies. So I kind of went over this slide a little bit, but it's important to assess and to think about all of these types of questions. What type of model to use? How much material do we have? What is our endpoint? Is it biologically translatable from one species to another? And all of these types of things before you select a model, because what I said earlier at the beginning of this webinar was that these are case studies designed to answer specific questions. None of these are one size fits all. These discussions, scientific thought being put into these beforehand, is the key to success of any of these studies. In terms of aerosol delivery, it's possible to deliver just about any type of aerosol you can imagine to just about any type of animal, whether that's a dry powder, a meter-dose inhaler, a nebulizer, a novel formulation, whether you'd like to do that in a nose-only setting, a face mask setting to a large animal, a small animal. It's always possible. It's just a matter of how you adapt it. What I've shown here is a very standard system on the left-hand picture here of just a rodent nose-only system that's very common, and I'll actually use that type of exposure as one of the examples that I'll show as we go through some of the case studies. Here are some other very similar systems that have been adapted for either a meter-dose inhaler or for a dry powder generation to a non-clinical nose-only rodent system. Now, any of you that have talked to me about IT installation, you understand that when I say IT installation, it makes the hairs in the back of my neck stand up a little bit. But even though it's not always the best delivery system, as I said before, in looking at a program and scientific hypotheses and questions, there are times when something like an intratracheal installation or some novel devices that can be used to spray or emit an in situ generated aerosol into the lungs of an animal, that's the right method to use. I think most people are familiar with this method where you do a light bit of anesthesia before you instill a tube or some type of a sprayer into the lungs of an animal and then deliver that dose. One of the reasons that, here's a couple of quick examples of some of these, and I think most of you have known or used these or seen these in the literature. There's some other novel ones out there that aren't as well published that all of them do a very nice job in a fit-for-purpose study where you're designing to ask a specific question that is okay with some of the caveats of these devices. This is one of the reasons why it's important to understand what your delivery mechanism is and what question you're trying to answer. The slides here show lungs excised from a rat following different non-inhalation techniques to the lung with experienced technicians. So this particular technician has about 15 years of experience doing these types of things. And what I really want everyone to see on these slides and want to be able to convey is that while these methods, oral aspiration, IT installation, those types of things, they deliver the material to the lungs. There's absolutely no doubt about that. There's no questioning it. The homogeneity of that delivery is oftentimes not very good. So in considering a study, if this type of delivery is okay, yes, an IT delivery can work. Whereas if you've seen any of you have published, and I apologize, I don't think I have one in this slide deck, the nice homogeneous deposition from like a radio-labeled scintigraphy study you get in the lungs of any type of an animal, That, in contrast to these images with the dye shown here, gives you an idea of the differences in the deposition patterns and potentially in your local PD or PK endpoints that you might be looking at. Again, if those are the right things for a particular study, those are manageable and then they're acceptable and okay. All right, all of that said, now I'd like to move into some case studies where we actually worked with the teams at Capsugel, as Devin mentioned, over the past 10 to 15 years, to actually go through some of these. The first one I'd like to speak on is a rodent study where we did some pharmacokinetics and pharmacodynamic studies with an endogenous peptide called PYY. The high level on PYY is that it is well known to cause appetite suppression when delivered by multiple routes of administration. The problem statement that the teams at Capsugel and Lovelace looked at when we started on this work was that this is a peptide was delivered by injection modalities, generally sub-Q or IP injection in the non-clinical models. Our question, our hypothesis was, could we develop an inhalation formulation of this material, and could we use pharmacokinetics to design an efficacy study and show similar efficacy from inhalation versus IP or sub-Q delivery? So the first thing we looked at was an understanding of the pharmacokinetics following the sub-Q delivery route. So you can see here on this slide the pharmacokinetics that show largely very nice dose separation and dose resolution for different doses from the sub-Q route. And as I stated before, our hypothesis was if we matched these pharmacokinetics systemically, so delivering to the lung and matching systemic pharmacokinetics, would we see similar efficacy in terms of reduction in food loss as the endpoint as a measurement for appetite suppression? So the first step in that was to work with the teams at Capsugel to generate a novel dry powder of PYY for delivery in a nose-only system. Those teams did a really nice job. We outfitted that material into a dry powder nose-only delivery system shown here on the right of this image to conduct PK studies in rodents. So our PK data in systemic plasma, shown here in the slide on the right, shows that again we see a nice dose resolution between our different doses for the systemic plasma exposure, and that we have similar profiles. Now, until we overlay that in the slide in the bottom, and really that slide in the bottom is what I would like everyone to look at, is to say yes, when we look at the sub-Q dose, and I'm sorry, I think I forgot to mention that, the sub-Q dose at 0.1 mg per kg had also shown some preliminary efficacy in the appetite suppression models. So our goal was to try to match that exposure. And if you see within the noise of a non-clinical model, largely the inhalation dose of 0.2 mg per kg matches the systemic exposure of the sub-Q route that was shown to be efficacious. So at this point, we felt we had the scientific justification to move forward into an efficacy model to look at appetite suppression. And obviously, because I'm presenting it, it should be no surprise that we saw reduction in food consumption as an endpoint that we used for appetite suppression within this model to show that, yes, we could take this novel formulation developed for a peptide, match the systemic pharmacokinetics for a systemic indication in appetite suppression, and show similar pharmacokinetics. There's further details on this on the publication that's shown there in the bottom in Drug Development and Industrial Pharmacy, if anyone wants to dig into that more. So the next case studies that I'll talk about are some large animal studies. And when I go into large animal studies, there's some advantages to going into the large animal studies. Oftentimes they're more clinically translatable. They're a little bit more relevant sometimes. And there's also some really cool things we can do with the delivery of inhalation materials to these animals. Now, the other advantages are that you can oftentimes do these models non-terminal, so you can do crossover studies and get a more powerful study with smaller animal numbers because you're actually using the animals as their own controls. So this slide is something that I think is very important and we all need to take for granted when we look at the differences between the model that we use and what we're actually trying to replicate in a human situation. In a perfect world in a human situation, most often a package insert says that a patient should deliver a material, whether it be a dry powder or a meter dose inhaler. I've got a dry powder shown here, in a nice, calm, usually 1 to 3 second inhalation where the patient fills their lungs completely and then does a breath hold. Now, there's some subtle differences in many of the package inserts out there that change this, but in general, that's what typically is supposed to happen. Hopefully, most of you are smiling right now and saying to yourselves, well, That never happens in a clinical setting. And you're probably right. Very seldom does it ever happen. So the ability to take an animal model and try to replicate that type of a maneuver in an animal model for a clinically relevant delivery, and if we can't do it in the clinic, how in the world can you do it in a non-clinical model? Well, long before I got to Lovelace, some folks developed a method that I'd like to talk about here to show some of the ability to do some clinically relevant maneuvers in non-clinical large animals. To start out with, the first thing we do to accomplish this maneuver is we anesthetize the animals lightly with isoflurane. Then we intubate the animals with an endotracheal tube. Then we connect an expansion holding chamber and a delivery device that it can either be a meter-dose inhaler or a dry powder reservoir, and connect that to a clinical ambi bag. What we do prior to the delivery of the aerosol is we induce apnea in the animals. For the non-biologists out there, that means that we over-oxygenate the animals and they stop spontaneously breathing on their own. What that allows us to do is it allows us to aerosolize these materials into the holding chamber, the expansion chamber, a split second before we deliver a breath to the animals with the positive ventilation or the ambi bank shown here. And after that delivery of the aerosol, we're able to do a simulated breath hold with the animals. Usually about 3 seconds, maybe a touch longer, so it can't quite be a full 10 seconds. But we believe this gives us one of the best opportunities to deliver a clinically relevant maneuver of an aerosol to a large animal. So now I'd like to talk through a couple of studies where we use this type of data or this type of a delivery to deliver materials for some case studies in large animals. So the first case study I would like to present and like to talk about a little bit is some work that we did with the teams at Capsugel to actually try to quantify and differentiate some of the tools they have to make different crystalline or amorphous materials in a PK/PD setting in large animals, where we're using local inflammatory markers as our PD endpoint and systemic pharmacokinetics as our PK endpoint. The overall study design is shown in the bottom here, and we were looking at these at the inflammatory markers in bronchial alveolar lavage 24 hours after a challenge with LPS. So this program was designed to look at fluticasone. It was designed to use the teams at Capsugel to develop a amorphous formulation of fluticasone and a crystalline formulation of fluticasone and compare that to Flovent PMDI. Our working hypothesis on this was that we could see marketed differences in pharmacokinetics based on the physical form delivered to the lungs. And our question that we really didn't know the answer to was, would we see differences in local efficacy when systemic pharmacokinetics were markedly different? Really from this slide, what I want to show everyone is that we gave similar doses of the fluticasone material as we did with Flovent. We did a little bit of a Flovent dose response, and we compared some IV data with fluticasone to be able to compare with an IV for some of the pharmacokinetics. So with regards to the pharmacokinetics, what I'd like everyone to be able to really focus on from these slides is that we do see a marked difference in our systemic pharmacokinetics following delivery of the amorphous, the crystalline, and the Flovent materials. This is a systemic measure of exposure. So now our question from the scientific teams was, what does this mean in terms of local efficacy? Let's skip over. This is the analysis of the pharmacokinetics to show that there were actually differences when you look at Cmax and that there were differences in the AUC when we analyzed these. So theoretically, we might expect that we would see differences in the pharmacodynamics because one of our classic measures for exposure, systemic pharmacokinetics, showed that we had differences. That said, when we got to our total cell numbers, when we looked at our inflammatory cell markers from bronchial alveolar lavage in this model, we did not see statistical differences between any of the formulations, even though we saw differences in systemic pharmacokinetics. All of these formulations were efficacious. Now, if some of you are thinking in the back of your head, wow, you might have been too high on the dose-response curve for fluticasone propionate in this case, in hindsight, and there's always hindsight in almost any study you do, We agree with you. We think that might have been something that could have contributed to this. That is why the actual last arm of the study we did, we reduced the dose of the fluticasone from Flovent to see if we saw a change in that. And as you can see, we might have seen a little bit of response in that dose-response curve, but we still see efficacy. So if that's your critique, I agree with you. We tried to address it, but it was not absolutely perfect, as in just any of the models as I started. They're a model. They're not always perfect. The last study I'd like to talk about is from a case study from the Capsugel teams that we worked with here at Lovelace, was with regards to another biological that should be familiar to most of us, which was a comparison of Exubra, the former inhaled insulin product from the Nectar Novartis Pfizer teams, where the teams at Capsugel developed a novel insulin formulation to evaluate in a PK/PD model in dogs. And so the slide that I have in front of you here, in order to make sure that we could accurately compare any of these materials to each other, we had to make sure that we were delivering the exact same dose. And I should have said this probably earlier on, that with the way we measure dose and the way we compare dose uniformity across any of these is we characterize the amount of material that is emitted from the end of the endotracheal tube. That is what's presented to the animals, and so any dose or normalization that I talk about has to be talked about in that terms, and that's how they're characterized. This slide right here shows that credit to the teams at Capsugel when they developed this material, knowing that Exubra was a very good product in terms of its aerosol performance and aerosol properties. The teams from Capsugel had a slightly more efficient in our delivery system, delivery efficiency than the Exubra product. So we had to do some mild adjustments to make sure we gave the exact same dose in terms of our loading and our delivery. In terms of the biological model with the PK/PD, this is well written up, and I've got the publication later on. This is the model where we actually utilize C-peptide, or sorry, we actually suppress endogenous secretion of insulin so we can compare these products to each other. So what I'm showing here is that C-peptide is dropped, indicating that we have suppressed endogenous insulin production, and the next slides that I'm showing you are fair to actually interpret their results. And so as you look through these, really what I'm showing you is that the performance of these material when we gave the same doses were almost identical. On this slide you can see there's a slight trend towards potentially some increase from the Capsugel formulation. When we did the statistical analysis with the N numbers that we had, they were not significant. Largely these products, when given the same dose, show the same PK in terms of your arterial plasma concentration of insulin, and in terms of your PD. When you look across any of these slides, any of these graphs, the glucose levels and the infusion rate required to hold everything the same were identical across all of these, meaning that when given, these maintain the same PK/PD. We did some pharmacokinetic analysis on here, and you can see some of the slight trends that we saw graphically. They match when you do the tabular analysis, very much the same. There might be a slight increase in some of the things like AUC, based on that time course where it was a little bit elevated from the Capsugel product. However, it didn't really trend when we started doing the statistics just because of the variability within any of these models. And so this summarizes that and gives the reference if anyone wants to dig into more of the animal model, the surgical procedure that's involved in developing that animal model. It's all in there. But this is just another proof of concept study to show an ability to answer a problem statement and to address a biological delivery of a compound that's designed for systemic indications with the teams here. And so I think with that, I'll go through this slide and then hand it back off to Devin. But what we really have talked about so far is the ability to kind of develop formulations, evaluate clinically relevant animal models, and then transition back into human disease. And what Devin's going to talk about now is some ability to take some of these formulation and some of these tools and get them into clinically relevant formulations or devices or filling mechanisms to actually transition this over into the clinic. So with that, I'll hand it back to Devin.
Thanks, Phil. So jumping now through this kind of process and formulation map earlier and jump into encapsulation. So as Phil talked about different preclinical approaches, what we'll show is kind of some early phase and later stage approaches that we've developed through capital purchases or internal capabilities to kind of have this holistic offering. And so what's shown here on the drug product side is capsules. Certainly there's other BPI devices such as the reservoir blister-based devices that are certainly suitable for these spray-dried powders as well. But on the early phase clinical supplies, we have an Excipient 600. This is a pretty unique platform, in my opinion, that it has a gravimetric filling mechanism. So there's a 100% weight check of capsules in line, and the filling mechanism is just almost like a salt shaker that's tapped multiple times. So you really get really low doses, and also a nice dose range flexibility that's also sometimes critical for early phase clinical supplies. With the gravimetric filling mechanism, and what's listed there is about 200, 300 capsules per hour. That's suitable for early phase clinical supplies, but for later stage and certainly commercial applications, that's not going to cut it. Which typically you're going to a volumetric filling mechanism at that point. So what we've recently purchased is a drum filling volumetric dosator from Harrow Hoffinger, which has more of the higher throughput. Multiple rows of stations and capsules to try and handle the later stage applications. What's shown on this slide is Excipient filled material. I think this is a Trehalose Leucine placebo. A mean particle size in the 2 microns. Certainly one of our worst flowing or worst handling materials that we want to really challenge the system. What's shown here is 3 different fill weights, kind of common ranges we might see for a early phase clinical study of 5, 10, and 20 mg fills. You say active loadings of, let's say, 10 to 50%. Kind of get single mg doses up to maybe tens of mg doses. And very high yields throughout the process. Pretty minimal development work. Really, it's almost the same process conditions for each fill weight. It just takes a little bit longer typically. So really nice from just a development standpoint as well. Similarly, taking this to the larger scale encapsulator. So what's shown here is again almost that same placebo done during a site acceptance test with the machine at full speed. Basically just running this thing till we ran out of powder, but ended up with a mean dose with the volumetric fill that we had at about 12.5 mg still, and a really low dose relative to standard deviation. So I thought that was pretty impressive for, again, a powder that definitely handles pretty poorly compared to some of the other powders we can produce, and in an initial challenge for this machine. Likewise, as we hit on again and again, water content. So certainly during encapsulation, equally important. We don't want to ruin the product or during that last step in the manufacturing process. So what's shown here is again local control for small units like an Excipient in the middle bottom picture, or with the larger units. Local control is certainly possible. Usually at some point you just need to go to a full suite control. So in the suite that's installed, shown in the lower right, we can control the entire suite down to less than 10% RH for the really hydroscopic formulation. Going to the capsule device part of the map. So I'm not really going to talk a whole lot about devices. We try and make our powders to be a little bit device agnostic or optimize them for a specific device and certain delivery profile. But I'll certainly talk about capsules a little bit. So what's shown here is kind of more the dose range flexibility that a capsule can provide. So 5, 15, and 60 mg fills, and kind of a line through unity of fill mass and emitted mass. So pretty similar results even at a 5 mg versus a 60 mg fill in terms of the emitted fraction or emitted percent. Of course, at some point there's a practical limit of how much powder you can deliver in a single capsule or inhalation, which is certainly real out there. But just kind of want to show the dose range flexibility of capsules, coupled with what we talked about previously, a fill weight active loadings in the formulation. Water uptake, again, another very critical part of consideration. Partly due as some types of capsules can become brittle, especially gelatin capsules, so a lot of the more hydroscopic formulations would typically be put into an HPMC capsule, which is shown by the different puncturing conditions on the right. But also, the hydroscopicity or water transfer between a capsule and the powder can be pretty important. You can imagine, putting a 5 mg fill into, let's say, a 45 mg size 3 capsule. If the water equilibration and content aren't controlled, you can have water transfer. The powder could act like a sponge, and when you open it up 1 month later, now you have just a little brick in that capsule. So again, hopefully I've highlighted quite a bit of how important understanding the water content is throughout the entire process, with the incoming excipients and spray drying, and as well as in the final drug product through again the manufacturing or the capsule is a pretty important consideration. Product characterization, I think just these last few slides, probably most people on here are aware of the different types of analytical characterization required for aerosol testing. A lot of it is focused on particle size, both geometric and aerodynamic. And just kind of I'll conclude with, again, water content important, maybe in a more unique way here of controlling the humidity just from a static perspective and ensuring you have consistent testing conditions throughout the life cycle of the product. So here's just shown a DDU and an NGI put into a controlled humidity enclosure such that we can always maintain the same humidity and test environment during these rather sensitive mechanical tests, which do rely a lot on the individual and a consistent work environment. And so with that, we'd like to thank everyone for coming. And if there are any questions, we can address them with the last few minutes.
Thank you, Devin and Philip, for the excellent presentation. We'll now move into the Q&A portion of this webinar. Our first question is for Phil. Would intranasal delivery be more effective or just as effective with your compounds? In your earlier slides, you have demonstrated PK results intranasally on rats.
So I think that question, really, the answer to that comes back to you can't make a blanket statement for efficacy on any of these without understanding exactly what question you're trying to answer. In the rodent study that I presented with regards to the delivery of a biologic for a systemic indication by nose-only inhalation, first let's establish that with any of those rodent models, there is always nasal deposition in those rodents. There was probably about 7% of what was actually breathed into the lungs of those animals that deposited in the lungs of those animals and stayed in the lungs of those animals. That is what we assign and believe, hypothesize, contributed to the systemic exposure and the systemic efficacy. Now, would there be a chance for a different delivery route, for a different formulation to have similar efficacy? Very possibly, that could be the case. That was just not the focus of that study, and that comes back to really where I started on some of these things, of saying the important thing to do is to understand what hypotheses you're testing and what are the strengths and weaknesses of the models out there that you can use to test those hypotheses, and then matching that model up with the formulation that you believe you can use to do that, to understand your question.
Thank you, Phil. And unfortunately, that is all the time we have. Any questions that we're not able to address here live will be answered by our speakers via email. I would like to remind you that there is a survey available after this session, and we appreciate your feedback. Please also contact us via email at solutions@capsugel.com if you would like to discuss anything around Inhalation Delivery Challenge or a project that you would like our assistance with. We will promptly follow up. Again, I would like to thank Devin and Philip for this presentation. Thank you for attending our webinar.